Welcome back. Now I've called this module Evolutionary Genetics number 1, but that's a bit of a lie really, because there's not going to be any evolution going on at all during this talk. What I'm doing in this module is just revising for you the basic population genetics under conditions of stasis, which are described by the Hardy-Weinberg equilibrium. Now if you're really familiar with that basic level of population genetics, you can move right on to evolutionary genetics number 2, where we will see some evolution in action. But if you want a little bit of a revision of basic population genetics, then stay with me for a few minutes. So, imagine a population made up of individuals, and we're going to personify these individuals as the Jean family right now. So meet, first of all, Mildred Jean. There she is, looking very happy. And George Jean, also looking very happy. And of course, they're very much in love because they're nice people. And the result of that is their child, Enid Jean. You'll remember that organisms such as humans are diploids, and so every one of our chromosomes has a homologous pair, and I'm just showing one pair of homologous chromosomes here in this illustration. And on this particular chromosome there's a gene called gene A that we're going to be considering in this example. And in this family, gene A exists in two forms, or two alleles, allele A1 and allele A2. And we show them here. On George's chromosomes, he happens to be homozygous for allele A1, whereas Mildred has one copy of allele A1 and one copy of allele A2, so she's heterozygous. You'll remember that parents give one copy of each of their homologs to their offspring at random, and so Enid could have inherited either both A1s, because both their parents have A1s, or that she could have inherited an A1 from her father, but an A2 from her mother. And in this particular example, we're showing her in the latter condition, being heterozygous. Now, in the remaining illustrations, we're going to do away with the human interest here, although it's cute, and replace these little images with the boxes I'm showing you now, such that on the left-hand side you see individuals that are like Mildred, who are heterozygous for the two alleles at gene A, and in the middle you see individuals like George, who are homozygous, in this case for allele A1. It's not so cute, but you get far more of them on the screen at any one time, which helps. So moving on, let's think about this family and a lot of other families in a town which we're going to call Jeansville. Here they are, made up of individuals that are vary at gene A. Some of them carry allele A1, some contain allele A2, and some are heterozygous for both. And it's pretty much random. Now this population, if we count up all of the copies of A1 and all the copies of A2, it just so happens that they're equal in number, so the frequency of allele A1 and of allele A2 is a half, or 0.5. So that's the condition, and if we had more than one allele, it would be the same sort of equation, P1 being the allele frequency of A1, P2 being the allele frequency of A2, and so on for any number of alleles at that same locus. But if you add all of those frequencies together, they have to sum to 1. So, all the people in Jeansville, they pair up and they have offspring. So we're now going to consider the next generation of Jeansville. What's the frequency of genotypes in that next generation? Well, the genotypes are composed by the combination of the gametes from the male and female members of the first population, and we're making the assumption that they're mating at random. So the female gametes will either be A1 or A2, and the probability of that is half for each, because the frequency of the two alleles in the population is equal. And the same is true for the male gametes. And then if we combine the male and female gametes, using those proportions, we'll find out what the frequency of the genotypes of the offspring are, just by multiplying them. So, just for example, A1 homozygotes, like I've just illustrated now, their frequency will be 0.25 because it's the product of the frequency of the A1 allele in the female gametes and the A1 allele in the male gametes. 0.5 times 0.5 is 0.25. The frequency of the A2 homozygotes is the same for analogous reasons, but the heterozygotes, there are two ways of getting the heterozygote condition, either by having an A2 from the male or an A2 from the female. And so we have two lots of 0.25, which is 0.5. So the frequency in this population of the three genotypes is 25% for each of the homozygotes and 50% for the heterozygotes. And you can see why. Now we can generalize the mathematics that you need to calculate those genotype frequencies if you know the allele frequencies like this. So the frequency of the homozygotes, in this case first line A1 homozygotes, is equal to the square of the frequency of the A1 allele. Similarly for the homozygotes for the A2 allele, it's the genotype frequency is equal to the square of the allele frequency for the A2 allele. For heterozygotes, the frequency of heterozygotes is twice the product of the frequency of the two different alleles. And the frequency of all the genotypes adds up to 1. The same set of equations can be used with appropriate modifications if you have more than two alleles at a gene in a population. You might want to go away and calculate what would be the frequency, let's say, of heterozygotes in a population of this sort if the frequency of the A1 allele was 0.2 rather than 0.5. Maybe we'll come back to that at the end. So what we've just described is known as the Hardy-Weinberg state. It's so called because the people that first did the maths to calculate this. And what it states is that the allele frequency and the genotype frequency do not change from generation to generation, but can be described, the relationship between them can be described by the equations shown in the pale blue box that you've already seen. That's the stuff to remember. In the dark blue box, I've also pointed out that you will usually find in most textbooks different terminology being used for the same stuff that we've been talking about just now, where alleles are called A, big A, little a, Greek A, what have you. It all amounts to the same thing. The equations are equivalent. I've chosen the terminology I've chosen just because it's more generalizable and doesn't imply anything about dominance and recessivity. Now, this Hardy-Weinberg state is an ideal state, and it only applies to populations if certain assumptions hold for that population. What are those assumptions? First of all, the assumption is that there is no selection. That is, for example, it's no better if you're a giraffe with a long neck than with a short neck. You're equally fit to reproduce, to produce the next generation. So no selection. Secondly, that your population is infinite. With an infinite population, then the assumption we were making earlier on that the gamete frequencies is equal to the allele frequencies in the population holds because there's no sampling error. If you have a smaller population, then you get sampling error and there's going to be some differences at random between those two. Thirdly, that there's no migration in or out of the population, bringing in additional alleles from elsewhere. And finally, that there's no mutation, that the DNA sequences remain the same from generation to generation without any changes. And of course, you know that you need mutation for evolution to occur. So, under these four assumptions, there is no change to the allele frequencies in the population. They remain static, and the genotype frequencies remain static, and that means there's no evolution, which is kind of dull. What we might want to ask ourselves is, can we use any of those equations, monkey around with them, to try and figure out what might happen if there was some selection, or the population wasn't infinite, or there was some migration, or there was some mutation? What would be the effect on evolutionary processes? Well, I think you can already see that real life is unlikely to conform to these ideal conditions in potentially important ways. So any one of them changing should lead to some change in allele frequency. And changes in allele frequency results in, or is the same as, microevolution. And that's the raw material of macroevolution, the origins of species and the changes of species. So if we want to try and ask those what-if questions about if there is some selection, if the population is infinite, and so on, and you'd like to have a go at seeing how we can model how much selection is needed to produce certain effects, well, that's present in the Podule Genetics, Evolutionary Genetics number 2, which you should turn to right now. There are some self-test questions which you go and try out if you want to make sure that you're familiar and confident with the things we've been talking about today, including the answer to that question I just gave you earlier on.